no-hopers, dead ends, or maybe hopeful monsters. No matter where you go in classic ecological literature, unisexual vertebrates are seemingly derided and tossed aside as inconsequential in an evolutionary context. This traditional view stems from numerous attributes of a unisexual life history, perhaps the most influential being that they lack the possibility of genetic admixture and subsequent recombination, resulting in unisexual lineages burdened with low genetic diversity and weak adaptive capacities, leading to rapid extinction. As clones, all individuals will share the same effective niche space, making them highly competitive with their conspecifics. From this traditional understanding, it follows that unisexual systems lack sufficient diversity to adapt to a changing environment, which should limit both their spread across heterogeneous landscapes as well as their persistence following landscape-level perturbations, short or long-term. After all, as realized niche space dwindles, unisexuals will have to compete more strongly with each other, while sexual species can adapt to this change. While hybrid genetic origins may mediate such niche space limitations in the short term by expanding niche breadth, unisexuals will still be haunted by this impediment over long-term evolutionary timescales or during rapid habitat transformation. Contemporary research into unisexual groups has begun to question this conventional way of thinking. Several examples now illustrate that some unisexual systems can persist for extended periods. Often these groups are found to be paraphyletic, a result of multiple independent hybridization events between parental species, with each individual lineage still constrained in time and space. However, the presence of genetic diversity within such unisexual groups, though minimal, has been found to help prevent complete collapse. Therefore, for any independent lineage to persist, it would still seem that additional genetic diversity is necessary, else succumb to the loss of viable niche space or long-term mutation buildup. Long-term persistence of the group, though, must mean they have some impact on ecological or evolutionary trends of other species, and thus warrant investigation beyond the scope of simply being novelties of biology. Unisexual Ambystoma appear to have defied the inevitable. This monophyletic lineage of all-female nuclear hybrids has persisted for millions of years, earning the designation as the longest surviving unisexual vertebrate species. Originally thought to be gynogenetic, unisexual ambystoma require sperm from a suitable host species for successful reproduction. A more contemporary understanding, though, shows that sperm genomes can be incorporated into an unreduced egg, resulting in ploidy elevation, producing communities of mixed-ploidy animals with variable genome dosages. In theory, any given single host community can be associated with up to a maximum of 4 genomotypes, or discrete groups defined by the genome dosages from the parental species, and where each genomotype has their own genetic composition and niche specifics. Importantly, each host will have their own unique set of associated genomotypes, while intermediate genomotypes may be formed in the presence of multiple host species, resulting in a synergistic effect on the diversity of unisexual ambystoma communities. Not only does the unisexual ambystoma system provide us an opportunity to investigate the role of genetic diversity on long-term survival of unisexual populations, but variation at both spatial and temporal scales can also be investigated. Pelee Island provides a test case wherein these dynamics can be investigated. Situated in the middle of Lake Erie, Pelee Island has been isolated for roughly 4,000 years, providing no opportunity for immigration from mainland ambystoma populations. Human development in the last 100 years has dramatically terraformed the island, destroying large swampland expanses and replacing them with a matrix of agriculture, homes, and roads. Here, unisexual ambystoma are syntopic with 2 compatible host species, the blue-spotted salamander, A. laterale, and the smallmouth salamander, A. texanum. Given our understanding of the unisexual reproductive system shown previously, we would predict that sites with a single host would have lower unisexual community diversity, compared to sites with both hosts. Furthermore, sites that differ by host will have differing unisexual communities, with these communities having greater genetic representation of their local host. Lastly, sites where hosts have been lost will see a reduction in unisexual diversity, specifically a loss of genomotypes that favor the lost host. From 2015 to 2018, we collected both adult and larval tissue samples from Ambystoma salamanders across the island. For each sample, we used 6 microsatellite loci, high-diversity neutral genetic markers with alleles based on length rather than sequence. 4 were species-specific, 2 for each species, and 2 amplify in both species. Each sample is then assigned a genomotype based on max allele counts from species-specific loci. Using this strategy, samples could be identified as a host species or a unisexual hybrid. Based on our samples, A. laterale is the least common host, present at only 2 sites, one of which also had a texanum. For within site diversity, we use 2 metrics, genomotype count, S, and the Shannon's diversity index, H', to account for relative abundances. Per the first prediction, we anticipate site D3.1 to have the highest values for both metrics because both hosts were detected. At every other site, only one host species was found, or, in the case of B8.2 and 8.3, no host was detected. As per expectations, D3.1 was found to be the most diverse adult community, though D1.1 had a similar H' value. In larvae, we likewise see higher diversity levels at D3.1. While this site has the same larval genomotype count as other sites, it did have a notably higher H' value, suggesting that rarer genomotypes are more abundant. Across-site diversity was assessed by calculating pairwise Bray-Curtis dissimilarity values from relative abundances. Based on our predictions, we expected to find sites where a texanum is the exclusive host to be more similar to other texanum-exclusive sites than laterale-exclusive sites. This similarity would be represented as lower Bray-Curtis values, while dissimilarity is shown with higher values. Comparing adult communities supports our prediction, as site B3, the only laterale-exclusive site, typically had higher values than other site comparisons. This point is emphasized when viewing the results with a dendrogram, showing B3 dissociated from the other sites. Similar results are seen when looking at the larval unisexual communities as well. Once again, comparisons to B3 are consistently greater than any other pairwise comparison, which can again be seen using a dendrogram. Much like the within-site diversity analyses, this does not show us specifically how these communities differ. For that, we developed a metric to calculate the relative genome representation. Genetic representation in this study reflects the genetic input from a keystone host species. Effectively, this calculates the average proportional representation of a host within the unisexual community. In a community of all diploid hybrids, for instance, both L and T genomes would have a 50% representation, thus zero bias. If a community has more L genomes than T genomes, it would be L-biased, and vice versa in T-biased communities. Any community with a single host should therefore have more genetic representation from that host, meaning that B3 should be more L-biased, while the other communities should be T-biased, and that is what we find in both adult and larval communities. While site D3.1 does have both hosts, there are far more texanum, and thus the unisexual community is more T-biased. Historic larval data collected from 1984 to 1991, presented in a 2004 Kosiewicz report, were used for analysis of temporal changes. Of the 4 sites for which sufficient data is provided, 3 were resampled from 2015 to 2018, providing an opportunity to investigate temporal variation of host and unisexual communities. The same metrics used previously were used on this historic data, revealing dramatic declines in texanum populations at 2 sites as well as extirpation of laterale at 2. Across all 3 shared sites, H' notably declined. At the 2 sites where both hosts were present, H' was high. Site D6 is lower than others, despite both hosts being present. But, like D3.1 previously, historically D6 had far more texanum than laterale, and therefore the influence of laterale was likely negligible. However, at a temporal scale, the unisexual community at D6 does not appear to have significantly changed, while B6 lost laterale and shows a change in community structure. Furthermore, comparison of A2 to contemporary communities shows high dissimilarity to all sites other than B3, the one contemporary site with which it shares a common host, further supporting our previous prediction of host-associated community structure. Lastly, genomic representation in unisexual communities further emphasizes the shifts in unisexual communities. Both sites that lost laterale have unisexual communities that have become more T-biased. This is particularly noticeable at B6, where laterale and texanum were both represented by 5 or fewer individuals. The reason for genomic bias to decline at B8.1 is uncertain, but may be attributable to the fact that larval samples for the site are almost exclusively from 2016. Rate of genomic incorporation is lower in colder weather, so if 2016 was a colder year, fewer ploidy elevated larvae would be expected. Alternatively, that year may have had a poor recruitment potential, considering over half of the samples were genetic clones, raising the possibility that they were all from the same mother. Based on our results, it becomes clear that local hosts play a significant role in determining unisexual communities. Similar results have been seen in other hybrid complexes, supporting the idea that unisexual systems would require a variety of reproductive modes in order to maintain long-term viability of a lineage without constraints imposed by sexual reproduction. While Pelee only has 2 hosts, Unisexual Ambystoma are known to use up to 5 potential hosts in the wild, helping to expand their niche potential. Having a range of specialized niches across genomotypes, along with their ability to sample from a multitude of hosts, likewise helps to expand the potential range of these groups, providing them a buffer against small, regional habitat changes and helping to ensure long-term lineage survival. Presence of such complexes imposes hurdles on their hosts as well. With a greater diversity of genomotypes, more niche space is competed for, leaving host species populations more vulnerable to extirpation. Short-term survival of unisexual groups would have little influence on survival of an entire host species, but in the long term, the continual added pressure by unisexual groups with a high diversity of niche spaces would impose limitations on host species range expansion. In areas of overlap between 2 hosts, there is more niche space being competed for due to the greater diversity of unisexual genomotypes. Consequentially, this niche overlap by unisexuals may serve to reinforce niche separation of the sympatric host species. Add to this the competition for sperm and habitat fragmentation as seen on Pelee Island, and it follows that host populations could rapidly decline or become extirpated where these unisexuals are found, much like what we saw with laterale and texanum. And if the hosts disappear, so too go the unisexuals. Perhaps the role of these no-hopers is monstrously underappreciated. So that's it. Thanks for watching our presentation. This system can be incredibly complicated, and hopefully we didn't overdo it too much with detail. If you have any questions, please feel free to email me or message me on Twitter about it. That's all. Bye.